Welcome to Curiosity Kick, the podcast about ideas for curious minds with big appetites. I'm your host, Lee Delaney. Before we get into this week's topic, I want to wish all my listeners a safe and happy Christmas and New Year, and to thank you for your support with Curiosity Kick so far. I also want to thank the amazing guests that have contributed to the first 6 months of the podcast. This is the last episode of the year, but I'll be back in January with more fascinating discussions. On this episode, I'm talking crystallography and the coronavirus with Dr Sam Horrell. Sam is a structural biologist at the Diamond Light Source Particle Accelerator in Oxfordshire, England, and also contributes to our understanding of the structure of coronavirus with the Coronavirus Structural Task Force. If you want to give me the best Christmas present ever, then tell your friends about Curiosity Cake and give it a rating and review on Apple Podcasts or whatever podcast app you choose. Now get yourself a cup of tea, grab a fork and dig into a slice of Curiosity Cake. Curiosity Cake. Sam, welcome to Curiosity Cake. It's really good to have you. Thanks for coming on.
Thanks for having me.
So you work in and around crystallography. So can you tell us a bit about crystallography, how it works, and maybe even, I think a question for me to begin with is, what is a crystal?
That's a perfect place to start actually with what is a crystal. So a crystal is anything with a regular repeating lattice in 3 dimensions. And the crystal is actually really important to how we can collect data for crystallography.
Right. So what does that mean then, the lattice in 3 dimensions?
So if you imagine, I'm sure you've seen at some point the structure of sodium chloride, where it's just a cube pretty much with sodium chloride, sodium chloride, sodium chloride repeating in X, Y, and Z. So that's probably your simplest representation of a crystal. We can crystallize a lot of other stuff. So I in particular work on proteins, which I guess most people wouldn't really think of as a crystal. Most crystals you think of as sort of your diamonds and your rubies and stuff like that.
Right, okay. And I guess this is getting into something we just talked about on our quick Zoom chat there, of people probably think of Gwyneth Paltrow type crystal healing when they think of crystals. So should we clear that up before we get into anything else, that's not what it is and that's not what crystals are?
Yes, I can confirm I have never ever artfully arranged a piece of smoky quartz on top of a woman's head in order to relieve their headaches or anything like that. Although I do actually have a school friend whose mum is a crystal healer.
Oh, okay.
I haven't met her since I started in crystallography, but if I ever do meet her, I imagine it will be an interesting conversation.
Yeah, very much so. So you've not cured anyone of a broken heart or anything like that?
I'm sure I've consoled a friend at some point, but I never cracked out a crystal to help with it.
Okay. So you're talking there about crystallography. So what is crystallography?
So crystallography is a type of structural biology or structural chemistry. So there's kind of 2 broad types of crystallography. There's the small molecule one, where you get like your sodium chlorides and stuff like that. And then there's the type of crystallography I work in, which is macromolecular crystallography. So this is much bigger molecules like a protein. And effectively what we do is we crystallize our protein, and then we fire an X-ray at it. The X-ray produces sort of a beautiful spot pattern, and these spots are effectively representative of the structure of the molecules inside the crystal. And then we use some very complicated maths, which I have a passing understanding on, and we can calculate the original structure through, it ends up being sort of an electron density map, and then we can build a model into that of what our protein looks like.
Okay, so this sounds like quite a interdisciplinary kind of study? You've got the physics there with the X-rays, the biology, the chemistry that you mentioned,
Hmm.
and then the mathematics as well.
Yeah, it's definitely a lot of moving parts. So I I sort of work in the biology lab and on the beam lines, which is where we actually collect the X-ray data. So yeah, starting from the very beginning, you effectively pick a protein you're interested in, genetically modify some E. coli or insect cells or yeast cells, depending on the protein you're working on, get them to produce your protein, then you'll purify it, crystallize it, and then hopefully solve the structure. But just
Mhm.
having a crystal doesn't necessarily mean you're going to solve your structure. It can be a lot more complicated than that.
So throughout all of that process, then, are you working with people from those different backgrounds at each stage, or are you more hands-on throughout all of those stages?
There's a lot of collaboration. So at Diamond, where I work, there's obviously a huge team of engineers and software developers, and then there's people like myself, which help users. So it is a user facility. So we do
Mhm.
get people from all over the world coming to Diamond to collect data. Although at the moment they're all coming in remotely and controlling the synchrotron probably from their living rooms or something.
And Diamond is a particle accelerator.
Yes, the proper name for it is a synchrotron. So it's
Mhm.
I can never remember the exact thing. I think it's about a half a kilometer diameter, and
Mhm.
it sits out in the Oxford countryside near Didcot, a very unexciting place, apart from the particle accelerator. But yeah, it's actually not a circle. It's 52, I think, straights, and every bend you have magnets all around that make up this ring. And whenever
Mhm.
the electron beam is diverted by one of these magnets, so it changes direction, it gives off X-ray radiation, and then
Mhm.
we focus that X-ray radiation with mirrors and apertures and all sorts of stuff to get it to a very small beam to hit our crystals at the other end.
From that X-ray beam hitting the crystal, you get a diffraction
Yeah,
pattern from that then tells you about the structure and nature of the crystal.
yeah. So, like I said, it's a lot of complicated maths to actually get to the
Hmm.
structure. But if you're just looking at the spot pattern, it can tell you some interesting stuff already just from looking at those spots. So,
Right. Okay. So the spot pattern then is the pattern that comes up from the X-rays hitting the crystal. It
yeah,
it's like a pattern of dots that are in different places and have different sizes.
yeah, I'm sure I can provide you a picture if you like to put up
Yeah,
with
that'd
the
be great. Yeah.
podcast. But
Yeah, definitely.
effectively, the spot pattern that you get is something called reciprocal space. So it's effectively the opposite of what is in the crystal.
Ah, okay.
If the crystal has really big molecules that are that are spaced far apart, the diffraction pattern, all the spots will be really close together. And same for the other way around. If it's really small molecules that are packed really close, then the spots will be further apart from each other.
Okay. So would the size of the spots then tell you something?
Yes, it can actually. So in the process of crystallizing our proteins, we use a lot of salts and other small molecules.
Hmm.
And sometimes you will in fact just crystallize salt, and you'll pick up that crystal, put it on the beamline, hit it with an X-ray, and you'll just see an absolutely massive spot. And you'll be like, well, that's definitely salt. It's not protein.
Right,
And then
okay.
you go back to the drawing board.
Yeah. So that can maybe give you some idea of the complexity of the molecules you're looking at.
Yeah. So, I mean, obviously, the actual structural and functional information comes after the spots, but it's kind of a common saying in crystallography that this is still an experiment, the collecting of the data. and you need to pay attention while you're collecting your data to make sure it's giving you the best chance possible to actually solve your structure.
And then that process of firing the X-ray, getting those patterns with the spots, and then understanding your structure, how long might it take you to actually build an idea of what the structure of your crystal is?
I mean, that's very much going to depend on your protein.
Mm-hmm.
So I have worked with a relatively easy one during my PhD. The quickest I've ever gone from expressing it in bacteria to having a structure was about a month. But
Sorry, expressing it in bacteria, is that when you're going through the process of growing a crystal to study?
That would just be growing the protein. So you
Okay.
you get the bacteria to produce your protein, then you purify it, and then you crystallize it. So that process alone on some of the more difficult proteins, particularly membrane proteins, the proteins that are embedded in your cell membranes, there are people that will spend their entire PhD trying to get a crystal, trying to make it express well enough and purify it to a high enough level and high enough quantity that they can actually produce a crystal.
Wow, so that's like years of work just working on producing a crystal before you even then get into studying the crystal itself.
Yeah, that's kind of why, like in the old days of crystallography when it first started, it was mostly physicists that would do it, because before computers, solving your structure was effectively a lot of maths. You'd have tables of logarithms called Beaver-Lipson strips, and from that you would solve your structure on paper. So obviously that was much more of a physicist's game. But now that the major bottleneck is sample preparation, crystallography, at least in the macromolecular sense, has shifted a lot towards less physicists and more biologists working on that step.
And is that the role of technology, as you can build particular equipment that then allows you to not necessarily have to do all of that working out through the maths on paper?
Yeah, exactly. There are suites of software now that you can just download, and for the most part, if you've got good data, will work with very little intervention on your part. Sort of, you'll point it at your files, you'll type in a couple of variables, and you'll end up with an electron density map, and you can start building your structure.
And I think, does that mean as well, before you had that kind of equipment, you'd be building by hand those like 3D models of proteins and things like that?
Yeah, so actually I think it's in the Wellcome collection, there is a really nice structure of penicillin where it's in sort of a half-open box, and they've built a wire model, and then on the actual sides of the box, you've got where they've drawn the electron density.
And then when you get to that point of understanding the structure of a protein, what does that actually help us understand?
So a lot of sort of the structure of a protein is kind of scaffolding to actually get you to the active site, which is normally sort of the business end of your protein. And a lot of what crystallography has been used for in recent years is something called structure-based drug design. So if you know of a protein in a disease which goes wrong through some sort of random mutation, or maybe it's a birth defect, if you know that protein and you know its structure, you can design a very specific molecule that will fit into it and inhibit it or make it more active depending on what you're looking to do.
So that's maybe a good point for us to then get into the impact of crystallography then. So you've hinted at a bit there. It allows you to really look at developing medical treatments.
Yeah, so what I would say is the true healing power of crystals.
But that's in a much more kind of pharmaceutical form.
Yeah, definitely. I guess the elephant in the room is COVID-19, and there's obviously been a lot of work in crystallography done or being done towards trying to design an antiviral.
if we maybe come back to COVID in a minute, because obviously that'll be a big topic to cover.
Sure. Yeah.
But I think crystallography was used like in mapping out DNA and things like that. So are there what other kind of discoveries have we made through using crystallography that have been important?
Yeah, good question. So crystallography is a very well recognized field. So I think we're up to about 30 Nobel Prizes related to crystallography. So the very first one of those was Röntgen, who discovered X-rays, which I guess is kind
Okay.
of tangentially related to X-ray crystallography several hundred years down the line.
But
So yeah,
still seems very fundamental though.
yeah, pretty helpful. So that was 1885, I believe, when that
Okay.
happened. But then crystallography itself didn't actually come into being really until 1914.
It's quite a young science then.
Yeah, yeah. Although it did, in my lifetime of working in crystallography, it has celebrated its 100
Hmm.
100 years of crystallography. But yeah, von Laue actually also got a Nobel Prize, and he effectively got it for proving that things are made up of atoms, and he did that by crystallography because he took a copper sulfate crystal, shined an X-ray at it, and got diffraction. But then the next major development in crystallography was the Braggs. So this is a father-son science team who then actually figured out the maths of how to relate your diffraction pattern to a structure. So that's how that's sort of the actual birth of crystallography. But yeah, you alluded to DNA, which is obviously a very, very fundamental thing of our understanding of science. And although, I mean, it's kind of, I think a lot of people know this story now, but obviously Watson and Crick proposed the structure of DNA. They figured it out from the bond angles and how it must interact. And then from that, they figured out the double helix.
Hmm.
But it wasn't actually until Rosalind Franklin came along with the famous photograph 51. She she wasn't able to solve the structure of DNA from that picture, to the best of my understanding. And Watson and Crick did manage to solve it, and were obviously
Okay.
awarded a Nobel Prize for that structure.
Hmm.
But by that point, Rosalind Franklin wasn't actually alive anymore, so you can't award it posthumously. So
Right. So
kind
she
of one
probably
of
wasn't that mad about it then.
no, but there's plenty of people that are still mad about it, and quite understandably, because
Okay.
from all accounts, Watson and Crick didn't seem to give credit where credit was due.
So yeah, well let's get back to COVID then. So obviously I actually haven't talked very much about COVID at all on the podcast because I think there's obviously so much around about COVID. I think there's definitely an element of people getting a bit sick of COVID, isn't there?
Oh
Sick
yeah.
of lockdown and all of that. But actually, I think this is a really interesting kind of way to look at it from its, deep from the inside of the virus. So,
Mm.
yeah, tell us about what you've actually been working on around COVID.
Yeah, so there's been a pretty big push for producing antivirals, and two of the main targets are the proteases in COVID-19. So I don't know how much you know about the life cycle of COVID, but when it
Maybe just assume I know absolutely nothing.
okay, so when it infects a cell, it releases RNA because it's an RNA virus, and
Mhm.
within that RNA, it first produces a long polypeptide, so a polyprotein, which effectively means it's a big chain with lots of individual proteins.
Mhm.
And then it will produce, I think within that polyprotein, it's protease. And that protease will then chop up the individual individual proteins from that chain,
Mhm.
and those proteins will then form and produce a big complex, which will then allow the virus to replicate its structural proteins, which is like the big hull that you see with all the spikes coming out of it.
Mm-hmm.
So the idea is that if we can stop this main protease from being able to cleave those polypeptides, then the virus will not be able to replicate. So we'll effectively have a treatment. It won't be a cure. It won't stop people getting infected, but it will be just like if you get an infection and go down to the doctor and they give you an antibiotic.
Okay, wow. So you're kind of then potentially trying to block the process that the COVID itself goes through.
Yeah, exactly. We're trying to
Yeah.
put a block on its major reproduction step.
Mm-hmm.
And yeah, the way crystallography can help with this is, like I said before, if we know the structure of this main protease. We actually scientists actually got on this really, really quick, and I think within a couple of months, maybe 2-3 months, the structure of the main protease was released. And since then, there's been sort of a big drive to try and design these antivirals. And the way crystallography goes about doing this is by something called fragment-based drug design. So what we do is we have a library of small compounds, and we'll soak those compounds into crystals of the main protease. And then those small molecules will work their way into the crystal and bind to the protein. And then we solve the structure, we see how they're bound, and we can then, from those small fragments, build a bigger drug molecule. And then we'll go through the same sort of iterative process. We'll get some chemists to produce those new, bigger molecules, soak them back into some more crystals, see the structure, see how that molecule interacts with different parts of the protein. And maybe you'll be looking at it and you'll be like, oh yeah, if I change that carbon to a nitrogen, this will bind better to this oxygen that it's next to, or whatever you're doing. Through that, hopefully we'll be able to generate some viable antivirals.
So how close do you think we are to developing those antivirals?
Still a way off, if I'm honest. So the problem is, this is sort of the basic research side of it. So even if we do produce a large molecule that binds the main protease and stops it working, it's going to be a brand new drug. So it's going to have to go through all of the clinical trials then after that.
Yeah, so and this isn't like a vaccine, as you're saying, that this would be an actual treatment. So it's not stopping people getting COVID, but it would be an option as a treatment for when they actually had it.
Yeah, exactly.
So would it potentially then like have a positive effect on the symptoms they're experiencing of it?
Yeah, I mean, obviously not being a medical doctor, my opinion on this isn't the most valid, but I would imagine if someone, at the point now we're at where testing is so quick, like people are suffering from it and maybe don't even have symptoms, but they know they've got it. If you can then provide a drug which will, within on a good treatment, sort of 1 to 2 weeks, and then they're free of it. It could be a way of getting sort of back to normal life if you can reduce the people that have it that don't know by treating it.
Right. Okay.
It's obviously not going to be able to reverse, say, the large scale lung damage that they're seeing in some patients. But for people that have got a cough, and haven't sort of gone in that big downhill slope where they're needing a ventilator, maybe that's when an antiviral could be a good sort of intermediate. I imagine it would be the potential to reduce severe hospitalization would probably be the main selling point of it.
Yeah, which obviously is really important, isn't it? So is that a project then that has a lot of pressure to it? Obviously, because COVID is such a huge issue, an immediate issue, or do you feel that this is something you've got to be working on as quickly as possible?
I mean, I'm a bit of a tangential player in the actual COVID work that Diamond is doing. So there's a whole team. They're called the XChem department. So they have been doing this structure-based drug design for many years now. So they kind of have just shifted all of their gears towards their effort for trying to solve COVID, really.
There's a couple of other things I was interested in around the crystallography. What is it about X-rays in particular that's what you're using as the radiation?
That's actually a great question. So if you kind of think of a light microscope, so the lowest, like the smallest thing you can see with a light microscope is based on the wavelength of the light. So I think you can go down to like going down to sort of the level of a bacteria. So that's obviously microns in size. But to be able to see atoms, you need a much smaller wavelength to be able to see them. So X-rays are actually the perfect wavelengths to be able to resolve these atoms. But then the big problem with X-rays is if you put them through a lens, they won't really bend at all. There's sort of almost no way of using sort of a lens to focus it like on a light microscope. So that's where crystallography comes in, with the detecting the spot pattern and using the maths to then solve the structure. So if you imagine the spot pattern is effectively in a light microscope, the point where the light has hit the sample and has kind of gone off in two directions and is picked up by a lens, and then you focus it back into the image. So that's the maths step of that, the focusing the light back into the image that we can see. So that's why we use X-rays, and then that's why we need half a kilometer wide building to be able to solve a structure.
So Sam, do you want to tell people how they can find out more about your work or where they can find you?
Sure, you can follow me on Twitter, which is at Dr Horrell, Horrell, because no one can ever spell that when I say it. I think it's because I swallow a lot of it. Yeah, other than that, I've written a couple of articles for InsideCorona.net, which is the Coronavirus Structural Task Force website.
Oh, brilliant.
Some of them are more serious than other articles. I think my most popular one is was the coronavirus made in a lab?
Right, okay.
It most likely wasn't.
Interesting.
Some spoilers, but
big spoiler there.
Yeah. Yeah, other than that, I would say check out Diamond's website. They've got some cool stuff on crystallography, and also the Protein Data Bank also has quite a lot of educational material if you're interested in learning more.
Brilliant. It sounds really good. Sam, that was really interesting stuff. Thanks very much.
No problem.
I hope you've enjoyed listening to this episode. Remember to rate and review the show on your podcast app and spread the word. You can get in touch on Twitter or through the website. Have yourself a fantastic Christmas and New Year. Bye for now.
Curiosity Cake.